former lecture. So I will going to address those questions which you have raised in the earlier, and of course they will be addressed during the lecture. So these different types of vectors we are using, and how viral vectors are able to serve the purpose. But I am assuming that you have no problem in understanding that why there is a need to send a gene of our own choice in humans. Okay, if you have, I just want to let me just revise a little summary of our last lecture so that we can connect it with the current lecture. So if we have a problem in human gene, remember, if we have a problem in human, when I said human, that means trillions of cells. So if there is a genetical error in human, theoretically, it is not possible to fix it. There might be certain scenarios where we can use gene therapy in humans, and I will show you some of them in adult human as well. But in order to fix a genetical disease, there is only one place where we can try that stuff, when humans are present at a single cell stage. That would be called as zygote. You can ask me, and we will going to discuss a lot about that stuff. So if there is an error in there is a fault in our stars, there is an error in the nucleus, there is an error in the mutation in the genes. So we will never be able to get rid of that stuff. The zygote that information will going to be incorporated in every single cell of the body. So what about fixing that stuff? In order to fix that error, for example, there is an error. We can fix this error by two means. One of the means is that if that error is there, that means, what that means? That means a gene from that region, if there is a gene, will not be able to produce a functional messenger RNA, and that will not going to produce a functional protein. So that means the messenger RNA will not be correct, will contain an error. That means the protein will not be functional, and that will not going to give you a function. Because of this loss of function, that cell will not going to execute a normal phenotype, and the individual will develop a disease. So there are one way is that individual contains a big part of the genome. How about sending something, the correct copy of that gene, into somewhere else of that same person? So for example, if this is the copy of that gene, how about sending the same functional copy of the gene in the other part of the genome? How about that? That will going to put the correct copy here. RNA pol 2 will going to produce a functional messenger RNA. The functional messenger RNA will going to produce the functional copy of the protein, and that protein will be able to execute the function. So although the cell is carrying the erroneous copy, the cell is carrying mutational copy of that gene, but the cell will restore the function because of the presence of this extra gene here. Because the extra gene will going to produce messenger RNA, it will going to produce protein, and it will restore the function. That is the original thought behind the process of gene therapy. But as a researcher, as a scientist, you should know when we can execute this process. If we want to fix an error in individual, we can do this job at a single cell stage. So that means that that we should learn that how to deal single cell treatment, how we can take out the single cell, how we can deal with the single cell, how we will going to insert things in the single cell, how we will keep the integrity of zygote and the stem cells at that time. When we can do those experiments, what are the techniques which are required to execute those experiments? A lot of technical expertise you need to develop in order to fix the things at a single cell stage. And last but not the least, how we will be able to know that whatever the things we are doing is done, is executed. The correct gene is placed and started working as well. So all these are the litigities and complexities which you should know at the end of the chapter. That it's not about knowing this is gene therapy. No, it's about the complexities and how you can execute those things. You should know, understand about all those things. So what we are using in is the viral vectors. Viral vectors are you can consider as that we have viruses, we create virons, viral particles. We do create virus particle for that. I can tell you that we can create this virus particle. But the difference is that these viral vectors or viral particles, they are somehow impotent. You know, viral genome might be RNA, viral genome might be DNA. The viral genome contains several important genes, and those important genes are involved in the creation of virus associated proteins in the future. When the virus will find human cells, they will going to hijack the human machinery, and they will going to produce the virus related proteins and some other factors which enables the virus to replicate, to reproduce things. So virus replication depends on the presence of certain genes or on the execution of the function of certain genes. We actually delete those genes. We actually remove those genes to make the virus impotent. What that means, the virus is successfully able to penetrate the human cells like this process. The virus is successfully able to deliver the payload. In this case, the payload is what? It is the correct copy of the gene. There is an erroneous copy of the gene is out there. On both the alleles, the problem is there. That gene is not going to execute its function. So we deliver the correct version here, and that will become part of the cells. That will going to replace them. The presence of a correct copy of the gene within the genome allows the cells to have that function. And virus is a medium because they have a huge potential of penetrating. I talked about that stuff last time, that what is the special thing about those barriers? The barriers which we call as cell membrane and the barrier which we call as nuclear membrane. And viruses, they do carry the potential to penetrate those important barriers. Laptop battery. No, sorry. I have no idea. I think it should be done. Okay, sorry for this time. Okay, yes, sir. So what we are doing is that we are using and harvesting the potential of virus particles that they are able to penetrate. We are using their potential, and then we are installing the gene of our own interest. If you ask me that the virus have no idea whatever the stuff it is delivering, the answer is yes, virus have no idea. They're delivering whatever we are giving them. So we are using the potential of virus machinery to deliver the things of our own choice. So those viral vectors, like the coronavirus is like one of them, flu viruses, lentiviruses, they came from HIV viruses. They have the potential to penetrate humans comfortably. So this is a viral DNA. Look at that. This is a viral DNA coding viral proteins, coding viral protein, and we engineer the viral genome or viral DNA and incorporate the gene of our own choice. This red stuff is the gene of our own choice. This is the gene of our own choice. This part. So we create a virus particle which is containing the portions and the genes which belong to virus, as well as the genes which we want that virus to take with those proteins. And also we deleted those genes which allows that virus to replicate itself within the cell. So when the cell is infected, the cell receives the DNA, but that cell will not going to produce viral proteins. That cell will not going to produce multiple virus particles. That cell will only take and receive whatever the material we are sending. Because the virus has a potential to penetrate the cell membrane. This is the first big barrier. The second, it has a potential to penetrate the nuclear membrane. Okay. in order to see how the virus particle can or a similar sort of technique can be used to fix a certain types of error in adults. There was a company who developed I will going to use the YouTube for that thing. This is the time to see that video. Okay, you guys should be able to see that, I mean, by yourself, but how about looking the video with me? Okay, I will going to share my screen with you, and I will going to share that one animation of that video. I should open this thing before. I'm going to stop this share and I will share my screen once again. So I will be speaking and you guys will have to see the video for that thing. Okay, I'm giving you one example that how pharmaceutical companies use this concept to generate a solution of a problem. And later on,
Parkinson's disease affects millions.
Are you able to listen to the voice as well? Just let me know, okay?
Yes, sir.
Yes, sir.
Voice of the video, okay?
Yes, sir.
Okay, that's cool. Okay, let's see. And remember, Parkinson's disease, as you remember, cancer is an age-related disease. Parkinson's disease is another disease which is imminent, which will be there, and that is also a kind of age-related disease. That sooner or later we will going to lose the control on our motor neurons because of aging. So the company has came up with a solution how to restore the function of the neurons once again, which was lost over the period of time due to aging. And they see and they utilize viral vectors to do that job. And just see and feel that whether you are able to capture the job of that company or not. Remember, the name of the products are always different from the things they are using. वो कमर्शियल नाम होते हैं ना, तो आपको कमर्शियल नाम को भी जरा अपने दिमाग में रखना कि वो कौन सी ड्रग है जो वो इस्तेमाल करते हैं और कैसे होएगी, and then you will going to ask questions about it. See.
Parkinson's disease affects millions of people worldwide. The main symptoms are caused by degeneration of brain cells that control movement. These cells originate in the substantia nigra and communicate with the putamen and caudate. As these cells degenerate, they lose their ability to produce dopamine and to communicate properly with other cells. Current medicines improve Parkinson's symptoms for several years, but become ineffective as the disease progresses. An experimental biopharmaceutical drug called SARA-120 has been safely tested in over 60 people with Parkinson's disease. SARA-120 is intended to restore damaged dopamine cells and protect them from further degeneration. SARA-120 consists of a bioengineered viral vector containing only the gene for human nurturing, a naturally occurring neurotrophic factor. This viral vector serves as a carrier for delivering nurturing to brain cells. Neurosurgeons accurately deliver SARA-120 to the areas of the brain where neurons are degenerating. SARA-120 then enters the neurons, inducing them to produce a continuous, lifelong supply of nurturin. Nurturin is expected to restore the function of the neurons and protect them from further degeneration, hence potentially reducing the severity of symptoms and slowing disease progression. A new study is now underway to test the potential benefits and safety of SARA-120 in Parkinson's disease. For more information, please visit
G. Any question? Are you able to absorb that video comfortably?
Sir, वो nursing आता है.
Gigi, please.
What did they say? It produced nursing?
मैं आपकी बात ठीक से नहीं समझ पा रहा हूँ. आप क्वेश्चन फ्रेज करेंगी? Can you create a sentence so I able to get what you are speaking?
They said that the drug induced nersin within the neurons, like a lifelong supply.
Oh, okay. This is the name of the gene, nurturing. वो मैंने इसलिए कहा था शुरू में आपको बताया था. I told you that please look at the names and commercial name of the drug and the name of the gene. So there is a gene which is responsible to produce dopamine. Okay, that's one thing you should know. Am I right? Are you with me?
Yes, sir.
And that is responsible for the connecting one neuron to the other one. That's like a normal and healthy way of communicating between neurons. So over the period of time, these got degenerated. The connections got degenerated. So when we give a cell that particular gene, we send that particular gene. And how we are sending? We are sending by injecting the viral vectors right within the brain of that person. That's a special thing. Dopamine is not required in your arms and in your legs and in your body, in your stomach, in your belly. It is required where? In the brain. So that the neurosurgeon is required to efficiently deliver that drug, which is the viral vectors, those viral vectors which are engineered and they are carrying the gene, the correct copy of the gene, or extra copy of the gene, I must say, to inject that stuff right in the neurons, in the brain. So once the brain cells receive that viral vector, the viral vectors are able to penetrate the neurons and then will start giving extra function. And that extra function is what? They start generating connections. It decreases the Parkinson and enables the patient to have better motor control over the period of some years. That actually help people to live a little bit longer than normal. Okay, so one more thing I think we should try to
for most
understand.
of us,
If you're able to, grab that video as well, I will be happy. Okay, I will going to share one more thing with you. Just have it and feel that if you have any question, please do write and do ask the question. I will going to share one more video with you. I want you to feel and to see that whether you are able to absorb the concept without any problem. Here we go. Are you ready?
For most of us, vision comes naturally. We don't think about the process behind it, but vision is a process. Light comes through the pupil. It is focused by your lens and it is turned into signals your brain can understand by an amazing bit of tissue at the back of the eye called the retina. The retina is the busiest part of the body. Each cell is a light processing factory. The factories gather light and convert it into electrical messages that can be understood by the brain. The operating instructions for these factories are called genes. People with inherited retinal diseases have an error in these operating instructions. This error, called a mutation, may mean that some crucial part of the process just doesn't happen, resulting in vision loss. One missing piece can shut down the assembly line and eventually the entire factory. The goal of gene replacement therapy is to provide new instructions to keep the factory working. So how do we get new instructions into the factory? The biological world already has specialists in industrial espionage called viruses. Viruses infect cells by sneaking into the factory. They take over the factory by giving the cell's machinery new instructions. Scientists use these stealth skills to design new therapies for vision loss. The scientists remove most of the virus's own genes, then they put new instructions into the shell of the virus. Scientists then inject these biological secret agents into the retina. Gene therapy can be adapted to treat many different blinding mutations. It cannot repair a cell factory that has been destroyed. However, the first human trials have shown that gene therapy can repair the assembly line and improve vision. Scientists funded by the Foundation Fighting Blindness are working on gene therapies and other
in retinal disease as well. The thing which you have just the two examples, they are two examples in adults which you have seen. And I told you that it is not possible to fix the gene, fix the error in all the genes. The answer is yes. What if a gene is required in one tissue only? What if the gene is required at one place only? Think about the first example, when the dopamine is required only in the brain cells, in the neurons. What about the other gene? In case of retinal disease, that one gene, the loss of that one gene is causing trouble, is a vision loss. But that one gene is required only where? In the cells which are creating the retina of the eyes. So there is a lens here, and this is the retina of the eyes. These cells are not able to produce that function. These cells are not able to produce that protein. So that one particular gene is not required in anywhere else in our body. It's not required in the skin cells, not required in the hair cells, in the body cells, stomach, kidneys. They are required only in the retinal cells. So if we have a target, we have a tissue where we can try to fix and restore the function. So that is a special thing in using gene therapy in adults, that you have to inject, you have to design a strategy to inject that adenoviruses or viral vectors with correct genes to the target place. And what is the target place? The target place is the retina. You have to inject those things in those cells. And let's suppose if the efficiency is like 10% or 15%, usually the efficiency is very low. It is not easy to fix every cell over there. The doctor might have to repeat that activity again and again in the eyes, and the patient might be knowing that he will going to receive an injection in his eyes, and he has no idea that actually he is delivered with the vectors, viral vectors, which will going to add a missing gene in their cells. But since the efficiency is too low, like I can tell you, I mean, even 4 to 5% efficiency is too much in this case. But even if the cells are able to deliver that thing to even 5% of the cells, even that is not the conclusion. That is not the. So if you ask me the problems in the gene therapy, the technical things with the gene therapy is the efficiency. First problem is that you will never be able to send the viral vectors to every gene, to every cell. It's not possible. You injected here the adenoviruses. These adenoviruses will be able to penetrate few of them, not all of them. If they try to penetrate in all the nearby cells, but I can tell you most of the cells, they will be able to not to receive those viral vectors. Some of them will be able to receive those viral vectors. Those some of them who able to receive those viral vectors, we itself need to see whether they able to incorporate the genome or not, whether they will going to incorporate the external viral vector or not. This is the human genome of the eye cell. It contains many genes. Some of the places they have lots of genes. Some of the places they don't have any gene. Some of the places are called as gene desert. And here we go. This is the adenovirus which brings this particular correct copy of the gene. So now the next question is that whether this inserted gene will be able to find a place here or not. So that's another question, another bottleneck. So who knows that where this external gene will going to land? It might land in the gene which is already doing a very important job. It might land in the promoter region of another gene, disrupting the function of other genes. It might land into the vacant area. So who knows where it will going to land? The answer is no. Nobody knows. There is no clue. We don't understand and know that during the gene therapy where the viral payload will be incorporated into the host genome. But theoretically, 98% of our genome is like a non-coding part. That means they are not producing any protein. They are non-coding portion. They are not producing any messenger RNA which will be translated into protein. So we can theoretically assume that 98% of the chance is that the incoming payload will going to be incorporated between the spaces of the gene. But who knows, the problem might be there. These are the shortcomings and the problems. But also the non-coding part, there are, the euchromatin part. You might remember this term. There are heterochromatin regions. So who knows that this extra gene will going to be entered into an heterochromatin part. The heterochromatin region is supposed to be the region which is very compact away from the RNA pole 2, and the genes present on heterochromatin are not expressed frequently. So in that case, the incoming gene will not going to be expressed if it is landed into the heterochromatin region. I wish you remember all these things. These are the complexities associated with the viral delivery system. So if you are lucky enough, you might be able to get few cells who receive the correct copy of the gene. And also they will start producing the protein, and you will see the restoration of the function. Even after that, I mean, even if you, theoretically it looks very simple. I will inject and things will be done. The answer is no. Okay, somebody raise the hand, please ask the question. I have Aisha Javed who raised the hand. Please, जिसने हाथ raise किया है, kindly इसको पहले पूछने दें.
सर मैंने भी किया था.
आपने भी किया था. Okay, आप पहले पूछ लें. Sorry, मैंने देखा नहीं. One by one.
सर वो मैंने पूछना था कि रेटिनल सेल्स में माइटोसिस होती है, रीजनरेशन होती है कि नहीं होती?
नहीं, उनमें नहीं होती है. नहीं होती.
ठीक है.
जितने भी न्यूरोनल एसोसिएटेड सेल्स हैं ना, जो डिफाइंड फंक्शंस करते हैं, वी डोंट पुश उनका वो फेज में नहीं होते हैं, सेल साइकिल को अडॉप्ट नहीं करते हैं. जी आयशा, आपकी बारी है.
Sir, so in gene therapy we only add the correct gene, but we don't take out the faulty gene. So maybe the function will be restored when the correct product is being produced, but there is also a faulty gene in our DNA. So if that faulty gene keeps on producing the faulty protein, so won't that have a negative effect still?
Write it, and note down this thing as a bad stuff of gene therapy. कोई आपके बारे में, whenever somebody is asking and talking with you about the bad effects of the gene therapy, let them know this argument. But imagine now, somebody is doing the bad thing, somebody doing the right thing as well. But before, what was happening? Only the bad thing was there. So now there was a bad protein, there was a good protein there as well. So we might be able to restore the function. So it's a little bit better state than